In this video, we're going to be talking about membrane transport. Remember that the plasma membrane is where a cell comes into contact with its environment. It's what defines a cell. So when we're talking about transport across the plasma membrane, we're basically talking about getting stuff into and out of the cell. There are a number of different ways to categorize and organize the various types of membrane transport. One basic categorization is to look at whether or not energy is required. If no energy is required to actually move something across the plasma membrane, it's referred to as passive transport. It just happens. The cell's kind of passive about it. If energy is required, it's referred to as active transport. Active because the cell has to actually work to move that substance across the membrane. Part of understanding the difference between passive transport and active transport is understanding the idea of a concentration gradient. So when we're talking about membrane transport, a concentration gradient basically means that you have a difference in concentration of a substance across the membrane. So let's say we've got an ion. If you have, let's say, sodium, if you have a lot of sodium ions on one side of the membrane and only a few sodium ions on the other side of the membrane, you have a sodium ion concentration gradient, where you have a high concentration on one side and a lower concentration on the other side. I'm using these brackets to indicate concentration. Now, substances in a liquid or a gas have a natural tendency to move down their concentration gradient from an area of high concentration to an area of low concentration. This is just due to the intrinsic energy of small particles like ions in liquid or, say, perfume molecules in air. If you open a bottle of perfume at one side of the room, if it's strong enough, pretty soon someone standing on the other side of the room can smell it, and that's because the perfume molecules have spread out from the area of high concentration in the bottle to the area of lower concentration in the room. As those particles bounce off each other and kind of careen off each other, they just simply spread out, and so they become more evenly distributed. This is a process called diffusion. Particles just basically spreading out from an area of high concentration to an area of low concentration. If you have particles moving from an area of high concentration to low concentration, that's called moving down the concentration gradient, and it's going to be passive transport. If you have particles moving from an area of low concentration to an area of high concentration, that's moving against the concentration gradient. It's like trying to take all those perfume molecules once they're spread throughout the entire room and trying to stuff them back into the bottle. It's gonna take some work, and it requires ATP. It's active transport. The simplest form of membrane transport is called simple diffusion. In simple diffusion, it is a passive transport mechanism. There's no extra energy required. It's diffusion, so you have molecules moving from an area of high concentration to an area of low concentration. And it's called simple diffusion because those molecules pass straight through the plasma membrane, straight through that phospholipid bilayer. Not all molecules can pass through that phospholipid bilayer. Remember that there is that hydrophobic center that acts as a barrier between the inside of the cell and the outside of the cell. So only substances that can dissolve in that hydrophobic center can do simple diffusion. So we're talking about hydrophobic molecules like fats, and some small molecules like oxygen or carbon dioxide can do it. Even water can do it as well because it's small enough that it just slips in between the phospholipid tails, but only in small quantities. This diagram illustrates one point that I want to make about diffusion. Diffusion continues until the molecules are evenly distributed. Notice that all of the molecules up here on this side of the membrane, they didn't all jump down to the bottom, but they diffused across the membrane until they're evenly distributed, until they're found in equal concentrations. And even at this point, when they're in equal concentrations, you're still going to have some molecules moving across the membrane. It's just that the number of molecules moving into the cell is equal to the number of molecules out of the cell. So it's more like in equilibrium. Another type of passive transport is facilitated diffusion. Like simple diffusion, you have molecules moving down their concentration gradient, but facilitated diffusion allows the movement of molecules that can't pass through the phospholipid bilayer. So when you see that term facilitated, basically it means that there are proteins involved. Proteins that are helping out, that are facilitating that diffusion. Proteins that are embedded in the plasma membrane, if you remember, can function as transport proteins. And these transport proteins open a little hole in the phospholipid bilayer that allow hydrophilic molecules to cross through that hydrophobic barrier. There are both channel proteins that just function a little bit like if someone stuck a straw through that plasma membrane, and they have this watery channel that hydrophilic molecules can pass through. Channel proteins are often used for ions, although there are channel proteins that allow water to pass through the membrane called aquaporins. And they select what passes through that channel protein basically according to size and according to charge. So they're not incredibly selective. But usually they will only allow ions of a particular charge or size through. The other type of transport proteins are carrier proteins. And carrier proteins, they actually change shape, allowing molecules to pass through. So they kind of pick up molecules on one side of the membrane and then shift and release them on the other side of the membrane. And since they have to have this shape change, they are more specific. So usually carrier proteins only allow a single type of molecule to pass through them. Osmosis is the movement of water across a membrane. And osmosis is, again, another form of passive transport. Only now we're talking about movement of the solvent. With diffusion, you're talking about movement of the solutes. Osmosis depends on two things. The first thing it depends on is the solute concentration, and the solute concentration is referred to as the osmolarity. So osmolarity is the concentration of all solute particles. Use the brackets for concentration again. Concentration of all solute particles in a solution. If you look at these beakers, you see that initially there's a difference in osmolarity. The one on the left here only has a low concentration of solute particles. The one on the right has a much higher concentration of solute particles. So the one on the right has the higher osmolarity. The one on the left has the lower osmolarity. So that's the first thing that osmosis depends on. The second thing that osmosis depends on is the permeability of the barrier. Notice that this is described as a semi-permeable membrane, and if you're going to have osmosis, if osmosis is going to happen, that membrane must be permeable to water, so water molecules can cross it, but it has to be impermeable to solutes. So the solute molecules cannot cross that membrane. If the solute molecules could cross that membrane, you'd just have diffusion. You wouldn't have osmosis. It'd be the solute molecules redistributing themselves. But if the solute molecules can't cross, but water can, it's the water that redistributes itself. And that's what you're seeing here. Here you have the difference in osmolarity. The solutes can't cross, so the water shifts. And you have now, after osmosis has happened, you have a greater volume of solution on the right because water has passed into this chamber. But now the osmolarities are equal. So like you're seeing in this example, water is passing from the solution with low osmolarity to the solution with high osmolarity. Now, the amount of water that is in a solution is inversely proportional to the amount of solutes. So the more solutes you have in a solution, the less water you have proportionally, and vice versa. Fewer solutes means more water proportionally. So in a way, water is moving from where there's a lot of water to where there's less water. That's the same thing as water moving from where there is few solutes to where there are a lot of solutes. One other idea that I want you to be familiar with is the idea of osmotic pressure. That's something that you'll encounter a lot as you're studying anatomy and physiology. And osmotic pressure is the pressure that drives osmosis. But when you think pressure, don't think like pushing on something. Think negative pressure, like a vacuum sucking stuff up, or sucking up fluid through a straw. The solutes in a solution exert osmotic pressure. So over here, where you've got a lot of solutes, this solution has osmotic pressure, and it's sucking the water across. It's sucking the water across that semi-permeable membrane. And that sucking, that's what's referred to as osmotic pressure. There are also some terms used to describe the relative osmolarity of two solutions. In this case here, where we're looking at red blood cells in a solution, the two solutions we're comparing is the watery solution inside the cell and the solution outside the cell. Now, to keep these three terms straight, make sure you look at the prefixes. The prefix iso means equal. So if a solution is isotonic to a cell, it means that that solution has equal osmolarity to the cell. And you will have water moving into the cell, but you'll also have water moving out of the cell. And there's no net movement of water, because the amount moving into the cell is equal to the amount moving out of the cell. If a solution is hypertonic, when you see that prefix hyper, think higher, like a kid that's hyperactive, it has too much energy. So a solution that's hypertonic is a solution with a greater osmolarity, or a higher osmolarity, meaning that that solution has more solute particles than the inside of the cell, and that water is going to be drawn out of the cell into the surrounding solution with its higher tonicity. And the net effect is actually a process called crenation, and crenation just basically means shrinking or shriveling. As water is drawn out of the red blood cells, they shrivel up. The prefix hypo means below or lower. Like if you have a hypodermic needle, it's a needle that goes in below the skin. So a hypotonic solution is a solution with a lower osmolarity. So fewer solutes in it than the cell. Water in this case is drawn out of the hypotonic solution into the cell. And you get if you let it go for long enough, you get lysis, and lysis is just a bursting of the cell, basically. The cells cannot contain cannot contain the water that's coming in, so they just rupture and burst, like you're seeing there. Okay, so we have our three forms of passive transport: simple diffusion, facilitated diffusion, and osmosis. Now I want to get into talking about active transport. Active transport can be further subdivided into primary active transport and secondary active transport. Primary active transport is usually transport of ions, and that's where you have a direct breakdown of ATP, driving movement of those ions across the membrane. Secondary active transport uses concentration gradients that are set up by primary active transport in order to drive movement of another molecule across the membrane. Okay, I'll give you an example in a minute of what I mean by that. But when you see secondary active transport, think 2 steps removed. So you're not immediately burning the ATP, breaking apart that ATP, but you set something up. You set up a concentration gradient through breaking down ATP, and now you're going to use that concentration gradient to move something else across the membrane. The sodium-potassium pump is a really ubiquitous example of primary active transport. Sodium-potassium pump maintains sodium and potassium concentration gradients that are absolutely essential for the functioning of your nervous system and for muscle contraction. Over here, you can see the concentration gradients that we have. So this is the extracellular side of the membrane. This is the intracellular side of the membrane. There is typically a higher concentration of sodium outside the cell and lower concentration inside the cell. And potassium is reversed. So potassium, there's a higher concentration inside the cell than outside. And the sodium-potassium pump sets this up because it moves potassium in to the cell, and it moves sodium out. So the sodium-potassium pumps in your cells are just constantly working. They're constantly picking up sodium inside the cell and moving them out, and they do 3 sodium at a time. And kind of as an exchange of that, they pick up potassium outside of the cell and bring it to the inside of the cell. Only they pick up 2 potassium at a time. So they're exchanging 3 sodium and 2 potassium, and they break down ATP in order to do this, going from ATP to ADP. So it's primary active transport. The pump, the actual molecule that is transporting sodium and potassium, is the molecule that's actually breaking down the ATP. And just as a good rule of thumb, if you see this in the future, when you see the term pump, think active transport. The sodium and potassium gradients set up by the sodium-potassium pump can be used by other molecules to carry out secondary active transport. This diagram is showing you an example of glucose transport across the membrane as an example of secondary active transport. So over here we've got the sodium-potassium pump moving sodium outside, moving potassium in. So you've got the sodium gradient set up, established, just like we talked about in the last slide. Now, there is a molecule, and it's called a symporter because it's bringing glucose in the same direction as it brings in sodium ions. If they're moving in opposite directions, it would be called an antiporter, but because they're moving in the same direction, it's called a symporter. And this protein brings in both sodium ions and glucose molecules. Now, sodium is moving down its concentration gradient. So for sodium, this is just sort of facilitated diffusion. But glucose is being brought in against its concentration gradient. Glucose is being brought in from the outside into a space where there's a higher concentration of glucose. So for glucose, this is secondary active transport, because this glucose is just coming along for the ride with sodium. This molecule that's moving the glucose, though, isn't actually breaking down ATP itself. It's just using the sodium concentration gradient that was set up by the sodium-potassium pump over here. One last category of membrane transport. It's a type of active transport, but it's different than what we've discussed so far, and this is called vesicular transport. It's transport using these little membrane-bound droplets called vesicles. It's sometimes also called bulk transport because a cell can use it to transport large molecules or large quantities of molecules. Here you're looking at an electron micrograph of vesicular transport. You have some individual vesicles within the cell, and you have some vesicles that you can see that are in the process of fusing with the plasma membrane. So here's the plasma membrane. Here's a vesicle that's just beginning to fuse with it, and here's a vesicle that's already almost completely fused with the plasma membrane. A cell can use vesicular transport to either remove things from the cell or to take things in from the outside surrounding environment. Endocytosis, when you see that prefix endo, think in. So endocytosis is particle uptake, so when a cell is taking particles in. Exocytosis, when you see that prefix exo, think out, or external. So exocytosis is particle secretion through vesicles. So actually I described this process as though it were exocytosis and the vesicles were fusing with the plasma membrane and spewing out their contents, but this could just as equally be an image of endocytosis, in which case what's happening, if this were endocytosis, what's happening is that these vesicles are budding off from the plasma membrane into the cell. You can also have what's called transcytosis, and this happens like in the cells of your intestinal epithelium. So transcytosis is where you have endocytosis on one side of a cell. So you have a vesicle forming, and then that vesicle is transported through the cell, and exocytosis, its contents, on the other side of the cell. So it's called transcytosis because it's transferring something across the cell. Exocytosis is really pretty common. It's used in a variety of cases. Hormones are oftentimes secreted by exocytosis. Neurotransmitters, so when cells of your nervous system are talking to each other, it's exocytosis of neurotransmitters. You also have mucus secretion, secretion of waste products. Exocytosis is really very common. With endocytosis, there are actually 3 forms of endocytosis. The first form is phagocytosis, and phagocytosis is only done by specialized cells. Cells of your immune system are really great phagocytes. Phagocytes is the term for cells that actually do phagocytosis. And these cells, they extend pseudopods, and that's what these little guys are, just extensions of the cell that wrap around large particles and bring them inside the cell. Pinocytosis you can think of as being cell sipping. Phagocytosis is cell eating, taking in large particles. Pinocytosis is cell drinking, cell sipping in smaller particles. And in pinocytosis, it's a routine process for most of the cells of your body. Cells just taking in little segments of the dissolved fluid around the cell. So extracellular fluid and any molecules dissolved in it, and breaking them off into smaller vesicles. A third type of endocytosis, receptor-mediated endocytosis, is less common, but it is a very specific form of endocytosis. So you have small particles interacting with specific receptors on the cell surface, and when you get enough of those small particles bound, the receptors cause this plasma membrane to pinch off, forming a vesicle. This is how iron is taken up by your red blood cells, as one example. All right, after studying this video, you should be able to answer these questions. Pay particular attention to the categorization and distinguishing between the different types of transport. So the types of vesicular transport, the different types of endocytosis, simple versus facilitated diffusion, passive versus active, primary versus secondary active. Make sure to keep those straight.